All right, so today I'm going to be talking about pseudogenes. So this is meant to be a part of the functional annotation channel, because if you're dealing with genomic or metagenomic or transcriptomic data sets, you're going to want to be able to filter out pseudogenes from your analysis or from your phylogenetic trees, or you may just want to study selection on certain genes. So I guess that might fall under a different channel. So I did do a lesson for the population genetics and evolution channel where I talk a little bit about this kind of stuff, but I wanted to do a lesson that's applicable to functional annotation. So this lesson is going to be under that channel. So pseudogene prediction using Pseudo-Finder. Pseudo-Finder is a program that I've been collaborating on with a couple of people, and I think it's a pretty useful tool. So there's going to be a tutorial after this on how to use that tool. Okay, so what's a pseudogene? So a pseudogene is a gene that, if you look at the sequence, appears to be homologous or related to a gene, but if you really study that sequence and you compare it to reference sequences, you would notice there's something weird about it. And these weird things could range from missing promoter or missing start codon to a frame shift on premature stop codon. There could be missing introns. So this is actually just for eukaryotic genes. If you're working with eukaryotic sequences, you're going to see introns, and those might be missing. Or you might see a partial deletion. So let me actually talk a little bit more about what each of these things mean. So a pseudogene is basically a gene that's inactivated, and there's a number of ways that it can be inactivated, as I just mentioned. Some are obvious and some are subtle. So this is the basic anatomy of a gene. You have a promoter, and the promoter is what binds the RNA synthase that allows the gene to be transcribed into mRNA. So transcription starts here, and this would be your transcript. You would have a ribosomal binding site, start codon, your open reading frame, and stop codon, and then potentially some downstream ORFs if you're if you have a transcript that's composed of multiple genes. So the ribosomal binding site. So the promoter is the first thing that can go. Promoter is a relatively short sequence. So if a gene loses a promoter, then it might just not be transcribed, and in that case, the gene will become inactivated. If a promoter is intact and you have a gene that's transcribed, it's transcribed to the ribosomal binding site, start codon. If the ribosomal binding site's missing, this is going to prevent the transcript from actually binding to the ribosome, and that also prevents transcription. Right, so missing promoter, missing ribosomal binding site. Okay. However, if you have a promoter that's intact and the ribosomal binding site that's intact, the gene will still be transcribed and will still bind the ribosome. But if you have an alternative or missing start codon, for example, here I show an ATG suffered a mutation to a GTG, which is also a start codon, but it's a less efficient start codon. Alternatively, you could have some other mutation that actually removes start codon completely. So in that case, you might have a stalled ribosome where it becomes stuck on the transcript and it never really starts translation. If you still retain a start codon and the ribosomal binding site in the promoter, so a gene is going to be transcribed and it's going to bind to the ribosome and it's going to initiate translation via start codon. But if you have a premature stop codon, for example, here I show a mutation from a TAT to a THT, TAG. That's a stop. It's called a stop mutation, and what you're going to end up is a truncated protein fragment. which could be not useful to the cell, or it could even be toxic to the cell. You could have an insertion that leads to a frame shift that causes a stop codon that also truncates the gene. You could have an insertion that actually moves a stop codon out of frame, and then a gene really is longer than it should be, and that might also result in an inactivated protein product that could actually even be toxic to the cell. Or you could just have a partial deletion of a gene where you don't even have a stop codon, so you might just have a piece of gene that's missing. Right, so basically, just to sum up, there's lots of ways that a gene can become inactivated and become a pseudogene, and what PseudoFinder ultimately does is it is able to detect these subtle and sometimes obvious gene breakage events that occur. And the way it does that is it's a reference-based approach, and by reference-based, I mean that it relies on genes that it assumes are intact, and these could be genes that are in NCBI's RefSeq database, or it could be a custom protein data set that you provide the program. And what it does is it compares the length of the genes to homologs. It compares, it looks for gene fragmentation. So if you have two open reading frames that are both shorter than the gene is supposed to be, and they actually are encoded next to each other and they match the same gene in the reference data set, then PseudoFinder will infer that it's two genes because there's a premature stop codon somewhere in there. Another advantage of PseudoFinder is that it is able to compare genes and actually calculate DNDS values. So a DNDS value is essentially a measure of gene conservation. And actually, if you're interested in learning more about DNDS, which I won't get into in this lesson, you could refer back to the population genetics and evolutionary biology lesson, where I talked a little bit more about what DNDS means in terms of selection. But just briefly to summarize, TS is the rate of synonymous mutations per synonymous site, and DN is the rate of non-synonymous mutations per non-synonymous site. So basically, if you have a lot of synonymous mutations in the gene compared to number of non-synonymous mutations, that means that a gene is relatively conserved, because synonymous mutation is a mutation that does not result in an amino acid change, whereas a non-synonymous mutation is a mutation that causes some kind of change in amino acid sequence. So if you have really low numbers of non-synonymous mutations, or really low rate of non-synonymous mutations compared to the rate of synonymous mutations, then essentially means that a gene is being highly conserved. So if you have a high DNDS, that could indicate neutral or adaptive selection. So if you have a really high DNDS above 1, that means that you have a lot of non-synonymous mutations. You have more non-synonymous mutations than synonymous mutations. And what that could mean is that a gene is undergoing some kind of adaptive shift. So it's actually the amino acid changes that occur are actually benefiting somehow, and they're become selected for in the population. Or alternatively, if you have a high DNDS that's below 1, but that's still higher than, let's say, 0.2 or 0.3, then that might indicate that a gene is just actually neutrally drifting, neutrally evolving, which means that there's really not a lot of constraint on the amino acid sequence, and that could mean that the gene is not undergoing selection. So I realize here that I just I stole the slide from another presentation that I gave on my research, which is with endosymbiotic bacteria. And we see that a lot in endosymbionts, where when a bacteria enters a host-restricted lifestyle, it loses selection on a lot of its genes that it no longer requires. So we see that DNDS actually is elevated in some of the in a lot of the genes that are no longer necessary. So if a gene is undergoing neutral selection, that's going to increase the likelihood that it's going to become a pseudogene, because you might have some kind of mutation that inactivates the gene, and that might that will eventually result in the gene being deleted, which is what we see over long time scales. So,
Hey, Arcade, can I ask a question?
yeah.
You were talking about a low DNDS value being above or below, I think you said 0.2 or 0.02, right?
0.3, yeah.
Okay, is that like an arbitrary threshold, like just acceptable by the community, or is there some science to show that that's meaningful in some biological way?
I think it's fairly arbitrary. Really, what you would want to do is you want to, if you have a genome that you're interested in looking for pseudogenes and you have a reference genome, what I would do is I would compare the DNDS values between all of the genes and look for the average value. And then you could do some kind of statistical separation, like 2 standard deviations above a certain average DNDS value. In the case of my research, I use 0.3 because the bacterial clad, the Sedalis, that frequently infects insects and becomes an endosymbiont. There was a paper that came out a couple years ago, and they did some analyses, and they concluded that 0.3 is the optimal number that they wanted to use to sort of draw that line at what's a pseudogene and what's not a pseudogene. But I think it's different for different species and taxa. So I wouldn't necessarily use 0.3 if you don't really have an idea of if you're working with a different bacterial species in
Got it. So organism, or at least population specific then. So you got to do
yeah.
some research. All right, on your own.
Yeah. Like in my case, when I do genome-wide average DNDS values between one like a recently acquired endosymbiont and its closest free-living relative, I usually get DNDS values that average at 0.09 or 0.08, and then the standard deviation is about 0.05. So 2 standard deviations above that is 0.2, and that's fairly. I think that's fairly a fairly accurate number to use in my case, because if I look at the DNDS values of pseudogenes that have been predicted using other metrics like length and gene fragmentation, then the average DNDS value is about 0.2. So.
All right, thank you.
Yeah. Okay, so this is a schematic showing the workflow, the pipeline of PseudoFinder. Essentially, you provided with a file, a genome annotation file. So this is a gene bank formatted file. And what it does is splits the regions by type, by gene sequences and intergenic regions. So one of the big problems that we see in the symbiont community is that a lot of the gene prediction software don't really do well in predicting genes in these in genomes that have a lot of pseudogenes. So what we might see is really low coding densities where we see gene sequences, but we also see really big gaps in between genes, which is really uncommon for bacteria. So what PseudoFinder does is it extracts these intergenic regions, and it also blasts them against a reference database here. And it also blasts the gene sequences themselves. And then by analyzing the blast output, we end up with pseudogene candidates. So, for example, and this is highly customizable pipeline. So, for example, if you have an intergenic region that's 30 base pairs long or 29 base pairs long, you can add a flag and tell it to not search anything that short. Or if you suspect that you might see some remnants of pseudogenes in really short intergenic regions, you can lower the threshold, and then PseudoFinder will also blast the really short regions. And then if that region has a blast hit in the database, then it's assumed to be a remnant of a gene. And then you could also alter that those parameters. You can tell it that if there's at least 5 hits or 10 hits or 15 hits to something in the database to this intergenic region, then only then call it a potential flagged pseudogene. And then it also, as I mentioned, looks at the length differences. So if a gene is significantly shorter than the top 15 closest hits, then that might be called assumed to be a pseudogene as well. So you can also vary the threshold. You can say if a gene is less than 75% the length of a reference gene, call it a pseudogene, or you can lower that to 50%. So it's highly customizable that way, which is pretty nice. So after this, it joins the adjacent fragments. So if it found fragmented genes that are separated by stop mutations, it will stitch them back together, and it will produce a set of output files, which I'll go over in the tutorial. This PAML loop is the NDS part, or the selection part of the program, where it's optional. So if you have a genome that you want to annotate, and you have a reference genome that is closely related, then you can provide that reference genome, and it will blast the 2 genomes against each other, and it will then use muscle and palatinal to create codon alignments, and then using those codon alignments, it calculates the NDS values. And then if you provide a reference genome, then you basically opt in to have this whole pipeline and these results factored in to the pseudogene predictions. So you can set the threshold, the DNDS threshold, to be as high or as low as you want. So if you want to set it at 1, then only genes where DNDS is higher than 1 will be flagged as pseudogenes. Or you could set the threshold to 0.3. You could also set a high cutoff. So you could say anything that's above 0.3, below 1 is a pseudogene. And that's if you suspect that anything above 1 is not a pseudogene, but might be actually undergoing adaptive evolution. Okay. Right, so these are the output files. I'll go over these in the tutorial. So PseudoFinder will also create these nice plots for you. So when you provide a genome, it will basically create 2 circles for you, and these circle plots. And the inner circle, will have the blue-colored input genes, and then that actually separates them on both the forward and reverse strand. And then the outer line shows the red-colored pseudogenes. So you could see in this Sedalis percaptivus is a free-living bacterium, and it has a 5 megabase per genome, and it's a pretty low density of pseudogenes. There's some pseudogenes, but there's not a lot. You can see some red in here. And on the right, we have a genome of a recently acquired symbiont, which is thought to be derived from Sedalis percaptivus. So this is actually a cultured isolate that's a free-living bacterium, and we also have a genome of an endosymbiont that is really closely related to Sedalis percaptivus. So we think that this endosymbiont is actually derived from a close relative of Sedalis percaptivus. And you can see that it has a lot of pseudogenes because it's been evolving in the host environment, in the host cytoplasm for quite some time. And similar thing here. So we have Pectobacterium wasabi, which is a free-living cultured isolate. And there's a endosymbiont that's closely related to Pectobacterium wasabiae, which has a lot of pseudogenes. So these genomes basically have about 5 to 10% of their genomes as pseudogenes, which is pretty typical for bacteria. Bacteria aren't really known to have more than 10%. 10% is actually pretty high, but I think one of the reviews that I read stated 1 to 10% of bacterial genomes are pseudogenes. Pseudogenes don't really stick around because bacterial genomes are really streamlined. But not in these recently acquired symbionts, which have about 50% of their 50 or more percent of their genes pseudogenized. And this is another, this is not an output from PseudoFinder. This is actually just a plot that I made from the PseudoFinder results, where you can see that there's a huge proportion of genes, or a larger than average proportion of genes that are shorter than their homologs in the free-living relative. So these are genes and then the symbiont. Right, so and then fragmented genes are open circles, and then intact or non-fragmented genes are closed circles, and all the ones that are red are basically pseudogenes. So you could see that when I ran PseudoFinder, in this case, I set the threshold for DNDS as 0.2, so anything above 0.2 is labeled a pseudogene, and then anything below 75% the length of the gene is called a pseudogene as well. So you could see that visualized here. Okay, all right. So we're going to move on to the Jupyter binder tutorial.